Okay. All right. Hi, everybody. Welcome to the ABE I4 seminar. And we are very pleased to have today for us Dr. Sierra Young, Assistant Professor of Biological and Agricultural Engineering at North Carolina State University. She has worked on the use of robotics and automation for sensing and sense making in agriculture and natural systems, also human-robot interaction, and she also conducts extension work in the area of sensors and robotics for precision management. Before arriving at North Carolina State, she was working as a visiting scholar at the Ag and Biosystems Engineering Department at Iowa State University. And she received her PhD in civil engineering as a Department of Defense Fellow, NDSEG Fellow, with a focus on human-robot interaction for physical manipulation from the very great University of Illinois at Urbana-Champaign. So we would have loved to physically welcome you here back on campus, but please accept our e-welcome, I guess, and the floor is yours.
Great. All right, I'll go ahead and share. How's that looking? Good?
Yep, that looks great. I think we have the full presentation.
Okay, wonderful. Well, thank you very much for the introduction, and I'm glad to be virtually connected back to my graduate school alma mater. At present, I am at NC State University as an assistant professor in the bio and ag department there. My research focuses on robotics and automation. And so I'll go through a couple example projects that I've worked on the past few years in these areas. So first, to give a little bit overview and background on my group, we are the Digital Agroecology and Intelligent Systems Lab, also known as the DAISY Lab. This is an overview of kind of the approach that most of my projects and research take. So here, kind of in the core, this large circle within this diagram encapsulates sensing technologies and data gathering. So most of my research focuses on the development and deployment of robotic systems and other sensing technologies in the field. This includes precision agriculture and also other biological and environmental systems. And so resulting from the deployment of these technologies, we always gather quite a bit of data. So taking those data, transforming them into useful information, and ultimately using those data to inform decision-making. And so this is a three-pronged approach where really the focus is on the sensing technologies and data gathering. And so I like to envision or kind of give the view that myself and folks who I work with to take on the persona of a broken comb. And so what does this mean? Well, it means that we work across a broad range of applications. So my background is in environmental and civil engineering. So things like water resources and land management through more precision agriculture, precision livestock, and even more detailed phenotyping work. And through all these projects, you might focus on a robotic system or developing sensors and controls. And as you'll see here, when my studies focused quite a bit on actually studying human-robot interaction to make sure these systems are usable. And so this is that broken comb approach that keeps things quite interesting. And ultimately, the projects are motivated by some problem. So it's really exciting to see, okay, what problem exists and how can we develop technologies, leverage existing technologies, to ultimately overcome some of these problems and difficulties. So within the realm of agricultural and biological systems, collecting in-field data is a very challenging task. So many of these systems, have their own unique challenges, everything from environmental challenges, navigation, perception, and control challenges, and dynamic and changing environments. And so, traditional methods of data collection in these areas, very labor and manual intensive, and, don't necessarily always get you the frequency and quality of data that may be needed to answer important questions. The use, however, of unpiloted systems, small robotic systems, can really improve accessibility and help us collect a lot more meaningful data more rapidly and often with more ease. So this includes aerial vehicles, ground vehicles, as well as surface vehicles. So here's an example of a water sampling UAV platform previously developed by one of my collaborators and now at NC State. You know, most people have probably seen the ClearPath vehicles used for agriculture. And this is a fun recent example, this kind of unmarked, essentially, I think it's a spy vessel that recently washed up on an island, I believe in Scotland. You know, the size and scale appropriate to some of the surface vehicles that researchers use. So really these vehicles take a wide range of form factors, a wide range of sensors, and really do improve our accessibility in some of these harsh field conditions. So we use them as science tools, and we use them to acquire data at spatial, temporal, and economic scales that are difficult to obtain by more conventional or manual or in-situ data installation methods. And kind of to encapsulate this from the field robotics perspective, the 4D scenarios in which these platforms are useful, situations that are dirty, dangerous, difficult, and dull. And so kind of comparing small robots across these three scales to more traditional methods of, as an example, remote sensing, where the ideal case is really having that really high or close to zero spatial resolution, a high frequency revisit time, high temporal resolution, and the lowest cost per data point. Robots qualitatively provide a nice compromise between some of these other methods of data collection. So in this talk, I'm going to focus on 3 different case studies, kind of focusing on different areas within biological and agricultural engineering, and a couple different technologies to highlight the usefulness of these systems. So I'll talk about a project that used a hybrid robotics approach to aid in the collection of hydrologic data, also focusing on the use of hyperspectral imaging to enable high throughput phenotyping in the forestry industry, and also looking at some human-robot interaction studies and the advancement of remote aerial systems for physical interaction with the remote environment. So first, I'm going to talk about this case study focused on hydrologic data collection. So to do this, we're going to talk about an area on the other side of the world in South India, in which this catchment has been continuously and significantly drying over the past few decades. And this is significant because this basin, this catchment, used to be the sole provider of municipal water, urban water supplies for Bangalore, which is a very densely and highly populated urban area right on the eastern boundary of this catchment. And so in particular, this drying trend was really pronounced in the northern subcatchment, specifically the T.G. Holly subcatchment, which is our area of focus for this case study. And so researchers have long been invested in understanding the drying within this region. And so there are a variety of hypotheses as to why the drying in this region has been occurring. So environmental reasons include changes in precipitation, amount and intensity, increasing evapotranspiration due to increasing temperature, and then a couple of human generated or human caused reasons for the drying. So that includes fragmentation of the watershed through the construction of check dams, which are essentially barriers in streams and rivers with the intent of stopping flow and increasing groundwater recharge. The fourth is declining base flow due to increased groundwater extraction primarily for agricultural purposes. And then land use changes, so a shift from other crops to specialty or cash crops. In particular, there's been a really big increase in eucalyptus plantations in this area. And so in looking at all these hypotheses, the environmental data historically really don't show any significant trends that would cause the amount of drying seen in this region. So those have been eliminated. And what's been left are these human-induced changes of drying. But it's really challenging in an area such as this to exactly pinpoint the contributions of each of these items to the overall drying trend. And this is challenging because the changes in water use and land use in these areas are spatially heterogeneous, and also they change through time. And the flow records within the Arkavathy Basin, specifically the TG Halley catchment, are really insufficient for understanding how the drying has changed through space and time. So when we look to see how can we better understand these human impacted changes in the watershed, one way we can look is looking at watershed fragmentation. And so this is significant because there is a very large number of these structures that affect the connectivity of the watershed. And so in particular, we can look at the tank structures. And this is just the number of structures in that subcatchment alone. And so what a tank structure is, essentially it's an open surface reservoir that historically, hundreds of years ago, would collect rainfall, and it was always immediately adjacent to agricultural land. So this immediate tank area would fill, and it would be where this is a spillover elevation, and there's also a spread area in case of heavy rains. And so what these tanks look like, they look kind of like this. And so they are relatively small. Here we are standing in the tanks, surface storage structures, but they really are an attractive target for understanding the spatial change of water availability because they're located all across the catchment. And many of them are large enough to be seen in historical satellite imagery. And so with this, they give us insight into rates of change of water within these tanks through space and also time. And so the goal of this project was to be able to map the bathymetry and topography of these tanks and obtain a relationship between tank storage capacity, tank surface area, and also tank or water level within the tanks. And this is then we can relate these relationships back to historical remote sensing data and understand water availability. And you might notice from these images that some areas of the tank, which I'm standing in here, are completely dry. But some tanks have areas within them that are wetted or full. And so there was not necessarily a one-size-fits-all approach for conducting the tank mapping. So we took a hybrid robotics approach where we used UAVs to map the entire tanks to capture the dry region, and then a surface vehicle to map bathymetry using sonar instruments in the wetted region of the tanks. And so with this, we have a UAV generated surface and also USV generated bathymetry in which we interpolate and get a continuous bathymetric surface and ultimately fuse these data in GIS to get a complete characterization of these tanks. And what's neat about this is that while this was done manually with local scientists and researchers, by using a robotics and having a robotics assisted method, we can collect much more data in a much more shorter amount of time and ultimately map many more tanks. So this is just an example of what those data look like. You can see this is the immediate tank area, and then because they rarely fill, the tank spread area had been filled with vegetation. But here we have the relationship between stage or the water level within the tank and the storage volume. And we can see that by also including the surface vehicle data, we get a more accurate representation of the true capacity of these tanks. And so what's really interesting then about these data is some of these tanks have water level sensors within them. So we can then relate the bathymetry and storage capacity data to historical stage data and ultimately convert to storage within the tank and tank inflow. So we now have an idea of when tanks fill, and we get a better picture of hydrologic connectivity within the watershed. And as you can see, that quite rarely happens with tanks spilling over into each other. And so just pointing out that these tanks really represent one piece of the puzzle. As I mentioned previously, there are quite a few hypotheses and possible reasons for drying within this watershed. And so here we are, and this is a larger hydrologic study published this year, but our tank inflow data is really one piece of the puzzle, looking at all of these processes that contribute to stream flow decline. And so I won't get into all of these individually, but ultimately these data, in conjunction with many other methods for collecting hydrologic data within that catchment, we're able to understand that groundwater depletion due to increased extraction, likely due to agriculture use, was the most probable driver of stream flow decline in this basin. And so this work was interesting and important to now make better policy and management decisions in that region to hopefully remediate some of the potential problems due to drying of the catchment. So now I'm going to move on to the next case study, hyperspectral imaging for high throughput phenotyping of loblolly pine. So just to put this in context, when I talk about high throughput phenotyping, it's using precision technologies to gather data to ultimately improve breeding decisions with the goal of improving a set of agronomic traits. And so phenotypic characterization is one piece of plant breeding. Ultimately, this does get combined with genetic information, environmental adaptation data, and many others. But when we characterize phenotypes, it's better to do this in a high throughput way. So you want to characterize potential phenotypes such as yield, disease resistance, abiotic stress resistance, plant architecture such as height, stem width, biomass, quality, and collect these traits for a very, very large number of plants, and do that frequently, and then be able to combine these data with genetic and environmental data to ultimately help us make better breeding decisions. And so typically high throughput phenotyping uses technologies such as imaging sensors, as well as field robotic systems, UAVs, depending on the scale, if it's field scale or greenhouse scale. And the data can be image-based, they can also be numerical or tabular data. So depending on the traits of interest, different technologies serve different needs in this area. And so in North Carolina, this is really exciting because we have quite a few plant breeders at our university that provide the sole source of cultivars for our growers, because there isn't any private investment in breeding these cultivars. This is specifically for many specialty crops. A few examples are peanuts, sweet potatoes, tobacco. You even have Christmas trees, which is really fun. So there's a lot of really unique and diverse crops that North Carolina state breeders are working towards improving. And so being able to collect high throughput data faster and at lower cost is really impactful, especially because many of these breeding programs are really one of the only efforts for North Carolina growers. And so the crop, or I guess the plant species of interest for this project is loblolly pine. And so this is a species that is unique to the southeast of the United States. And it is quite dominant on 29 million acres. It provides a lot of revenue for the state of North Carolina, but a lot of states in the southeast, including Georgia, Alabama, South Carolina. And it's the wood commonly used for things like paper products and pulp and plywood and general construction. This work is in collaboration with the Tree Improvement Program at NC State. And you can see here, it's a little illustration of what loblolly pine looks like. It has a very tall, normally straight, pretty uniform trunk, which is ideal for timber production. And so there is a large program invested in improving loblolly pine for this region of the United States, and North Carolina has one of the oldest tree improvement programs. And one of the most important traits that they look at within this program is adaptation to minimum winter temperatures. And so here, I apologize, this is a pretty old figure, it's not the best resolution, but hopefully you can see these isotherm lines that indicate mean minimum temperature, and are kind of a guide for where you should or where it's recommended to plant specific genetic families of loblolly pine such that you have reduced risk of essentially damage and ultimately death of these species due to cold and freeze damage. So you can see that the northernmost line kind of goes through Virginia, but through North Carolina and Tennessee. And this is really the farthest north that it's recommended to plant loblolly pine for timber production. And so ultimately, one of the traits of interest is increasing the ability for loblolly pine to withstand extreme freeze events and extreme cold temperatures, such that we can push that northernmost line farther and farther up north and expand the region where loblolly pine stands are planted for expanded timber production. And so this is really one of the most important climactic variables that's related to growth, and it does guide the transfer of seeds. So you might obtain seed from this line right here, and they would say don't go farther north or farther south than 1 isotherm in either direction. So they are important bounds geographically, as well as related to expected plant performance. And so typically seed is characterized by a mean minimum temperature, and also which is informed by the mean minimum temperature of that family's parents, the genetic parents. And so you might expect that becomes easy to quantify and describe if you are crossing loblolly pine genetic families within, close relationship to each other between these isotherm lines, but they also perform what are called wide crosses, where you might cross a north and a south genetic entry in the hopes that you might obtain some of the cold hardy traits from that northernmost seed and also the fast growing traits from the southern family. But you can imagine then characterizing the mean minimum temperature or characterizing the actual cold hardiness of that cross becomes challenging, and even more challenging when you do farther and farther crosses and you have more and more and more parents. So this, in the context of tree breeding, their cycles are much, much longer. So I believe, a fast cycle would be something like 5 years. It used to take 10 years. And so the tree improvement program is I think less than 10 cycles in their whole breeding since the existence of the program. So it's definitely much slower, and consequently high throughput is relative, but they're always looking for ways to speed up the cycle, the breeding process cycle. So what's needed is really a method that's reliable and accurate and more high throughput to assess cold hardiness, and ultimately methods that are non-destructive. So previously you might plant a stand and watch them and observe them in over a couple of years, exposed to different freeze events. But we are seeing, can we subject seedlings, small plants, to an artificial freeze event and ultimately get an accurate assessment of their cold hardiness? So in this study, we're looking at the potential of hyperspectral imaging as a screening tool for cold hardiness in loblolly pine. And this is just an illustration of where the source seed for all of the families included in our study came from. So after obtaining, sowing, and rearing some of these seedlings, this study was done earlier this year. So back in January, we had a test done where we froze the seedlings artificially. We performed a pre-freeze scan and a post-freeze scan, and also completed visual scoring at the end of the assessment as would normally be done in a breeding program. So we had 95 families and 3 check lots. Each replicate had 98 seedlings, and we had 20 replications. So we had just shy of 2,000 seedlings in this study. 16 of those reps were frozen and 4 were a control group. And so they were all of the seedlings that were frozen were subject to what we call the big freeze, which was essentially a simulated extreme freeze event midwinter. So we took the seedlings from the greenhouse, put them in a temperature controlled freezer in our collaborator's lab, Dr. Livingston, and then moved them back to the greenhouse to allow the injury symptoms to develop. And you can see here that even before freezing, there's quite a bit of color variation. because going into winter, some of the seedlings kind of harden off and go dormant. There's color change associated with that. So there's variability kind of before even the freeze, and then this variability is essentially still present and exacerbated after the freeze. And so we collected data before the freeze and also multiple time points after, and then we did visual scoring a couple months after, as would normally be done, to assess how many seedlings had survived and recovered and how many had died, essentially, mortality rates. And we did the hyperspectral imaging with a visible near-infrared imager that had quite good spectral resolution. And we imaged the seedlings kind of vertically, so we get one entire seedling at a time in their tray. And this is just an overview of the data processing pipeline, where we start with our hypercube, 400 to 1,000 nanometer images. And one unique thing about these trees at the state they were in is that the normal vegetation indices for segmentation, such as NDVI or perhaps excess greenness, really didn't work quite well because the seedlings had a quite a big range of color difference. They had purples and browns and greens, which made it difficult to use kind of more existing indices. So we did come up with a new difference plants index for completing the segmentation based on the plant background separability of the wavelengths. And then we did automatic thresholding, plant segmentation. We separated out the individual seedlings. And then also we separated the seedlings into a top third, middle third, and bottom third. And this is important because after they are subjected to freeze damage, they recover, and growth usually occurs at the top of the plant. So we're also interested in seeing if there's a difference between the top, middle, and bottom segments of each seedling. And so ultimately we correct and average the spectra for the entire plant, and we have corrected mean spectra for all of those sections as well as the seedling in its entirety. With these data, we also have to make connections back to the data from the breeding program. So we have our reflectance data from this experiment and also a freeze damage and mortality score obtained from visual observation. And we also have the mean minimum temperature that the seedlings were characterized with, that their seed came from, which is, as I mentioned previously, a really common measure for loblolly pine species. So first, we look to determine, can the reflectance of these seedlings be used to predict whether or not a seedling is going to recover or suffer from mortality after subjected to a freeze event. And so just first looking at some spectral features and spectral ratios of the seedlings to get a little insight on their growth and performance. We can see here that the quote healthy or recovered seedlings are the dotted lines, and the stressed or damaged are the solid lines. And so we can see some definite trends of recovery where, it looks specifically for plant chlorophyll concentration and plant water concentration, these band ratios have been shown to correlate with these two. And so we can see recovery. In particular, I mentioned the top of the seedlings, so we can see that here it's really pronounced, as well as here for both chlorophyll and plant water concentration. So this is essentially an indication that yes, we can at least relate reflectance data to the health and stressed or healthy state of the plant. And so ultimately, can we use these data to make predictions? To do that, we did cost-sensitive linear SVM modeling. This is because we did have an imbalanced data set where we had more stressed plants than not. The freeze was pretty severe. So after obtaining this tunable cost metric for this model, we did modeling for the pre-freeze, this -6 day, for the 4 scan dates after the freeze, and also separately for the full top, middle, and bottom of the seedlings. We split the data set into training and test data, applied the model, and then assessed a variety of performance metrics, including geometric accuracy and accuracy using true negative, false negative, and true positive as calculated from the confusion matrices. And so just looking at the classification results, we can see, as expected, the farther the time point from the freeze, you get quite good accuracy for predicting whether or not the seedling has lived or is going to die, as expected. But what's quite interesting also is that, as we hypothesized, the top portion, so the second orange bar here, pretty much in every case besides one, had the highest accuracy for predicting healthy or stressed. And so that does indicate that the new growth and also the growth that happens after the freeze, which is important because we are scanning the seedlings from the side, but in the future, to do a top-down scan of an entire tray would make this much more high throughput. And one other interesting note to point out from these results is that the detection accuracy before the freeze was actually 75%. While this is not extremely high, up in the 90s, as a couple weeks after, this indicates that the state of the seedling in winter, after it's hardened off, has pretty good predictive ability to predict whether or not the seedling dies or survives after a cold event. So, we want to repeat this study, of course, but this is promising in that perhaps if we can tune this method, we don't even need to conduct the freeze at all. We can improve this model and ultimately get a sense of cold hardiness of the seedling without even doing that freeze treatment. And briefly, just seeing if we can relate reflectance data back to mean minimum temperature, which is that characteristic that is of critical importance. So to do this, we said, can we do family level rather than seedling level predictions of mean minimum winter temperature? So started with all of our seedlings. We excluded the wide cross families for this study. And we averaged the data at the family level. So we ended up with 77 seedlings per family. We have mean spectra of the top portion of the seedlings before the freeze. And ultimately, we're working on these data still, but preliminary results with just doing some regression models indicate that there is some predictability of the, again, the reflectance data to predict the source mean minimum temperature of the seed. So this is exciting and ongoing analyses. And ultimately, what we're looking to do is be able to select the most important wavelengths. For example, we found the top two at 699, 767, and also looking at the accuracy as a function of number of wavelengths to deploy this at field scale, looking at scanning a large number of seedlings top down. So that's the long-term end goal of some of this work. And so I will now move on to the third case study focused on remote aerial manipulation, some applications in agriculture. So we are probably all familiar with the use of unpiloted aerial vehicles for remote sensing. A lot of applications in precision agriculture, phenotyping, environmental research, using a variety of sensors. And this is very useful for obtaining information for many applications. And so this is how most of us have probably come to know what UAVs are capable of, remote sensing. Up until recently, really the past decade, we can now use UAVs to physically interact with the remote environment. And so over the years, we've seen these capabilities increase from simple probing or mounting to now grasping objects and coordinated object transport. The controls and capabilities of these systems have really improved greatly over the past 5 to 10 years. And so this is a really exciting area of research that I see a lot of applications in for, field environments. Up until now, most of this work has been done in a controlled lab setting for controlled reasons. But it's important to note that many of the lab settings have systems such that it enables autonomous manipulation. And when we take those systems into a real world, where perhaps the sensing and perception and feedback, where you no longer have markers and tracking cameras and there's environmental noise, the capability of those systems to do fully autonomous task completion is greatly reduced. And so for, especially in environments where you might not know exactly what task you need to complete, such as sampling or perhaps object retrieval or placement, the ability reduces even farther when the task is unknown. And so we can then look to say, how do we enable UAVs capable of manipulation to perform tasks in a real world, dynamic, unstructured, natural environment? And I would argue that that answer right now, at least, lies in improved human-robot interaction for these remotely piloted systems. And this really is taking into consideration the capabilities and cognition of a human, in addition to the capabilities and automation of the vehicle, and designing the system such that both capabilities are optimized. As I'll show on the next slide, this is also encapsulated in what is known as the shared roles model, originally developed for field robotics. And so we assume, that the unpiloted vehicle has some level of autonomy. The humans are operators, but also system monitors and decision makers, depending on that level of autonomy. And designing human-robot interaction is more than just the display. It's about the larger systems level design and the choices that we make in that design such that the system is usable. So when you look at human-robot interaction for remote manipulation with UAVs, this is really largely an unstudied problem in the context of focused HRI. So telemanipulation is a widely studied problem, but in the context of stationary manipulators or missions that are long in the course of hours, perhaps ROV, remote operated vehicles, underwater vehicles, have really long missions if they're tethered. Remote surgery is considered telemanipulation. A lot of these setups have large stationary dedicated control rooms where you might have a leader follower setup. When we look at doing some of these tasks from UAVs, now we have a mobile lightweight ground control station, typically an iPad. You can't really have that leader follower setup. You might use joysticks, but it's probably going to be a touch device. And now we have short missions. We don't have hours to complete a remote manipulation task. We have to do it in less than an hour. So as part of my research, I wanted to understand how can we most effectively enable a human operator to perform a remote manipulation task with a small UAV. And to do that, I implemented the shared roles model, where in this model, we have a pilot who is responsible for navigating and operating the UAV. And note that as the platform autonomy increases, this may be a person, but it also may take on the role by an autonomous software agent. But ultimately, the role of navigation and piloting is delegated out, away from the mission specialist, who in this model is responsible for operating the payload. So typically, when the shared roles model was implemented, this was in search and rescue, field robotics, where they were doing visual inspection, perhaps searching for victims. But this really translates quite nicely to manipulation, where now that person is in charge of performing the manipulation task. And so they have complete control over the payload. And of course, there is verbal and other interactions between these roles. And ultimately, they must be connected in some way, but really informing that the tasks are delegated to better take advantage of those capabilities. And so in this study, myself plus a software agent took on the role of a pilot, and we had people who didn't really have much experience with UAVs come in as the mission specialists. So this was the model in which we conducted a human-robot interaction study with real participants and a real robot. And so the primary question for this study was, what types of views are best for a variety of tasks? So when you have a UAV, the payloads are limited and the locations in which you can mount sensors is also a bit limited. So there are two essentially different types of views. One is a more exocentric view where you have a complete view of the manipulation space. And the other is an egocentric view, where that view, sometimes called an eye in hand view for manipulation, is right up at the end effector and gives you a close-up view of the object or task. So we had participants operate an interface that had either the exocentric view, the egocentric view, or both. And with those views, each time they completed a series of 3 tasks. And these tasks were designed to be different and also have different difficulties. So the first task was simply a probing task where they had to touch the center of the target or touch anywhere on the target as close to the center. The second was a grasping task to grasp this hoop, drop it down. And then the third was a pushing rotation task where they had to lift up on this plate and it would snap to the magnets at the top. And then there would be random letters and symbols here that they had to then visually read out loud. And so these tasks were then performed by all participants with all views. So just as an example of kind of what this looks like, this would be the flight arena with myself, the pilot, and the UAV was station keeping autonomously. And then we have mission specialists, the participants who are faced away from the arena and did not have a direct view of the system. They could only view the system through their interface. And so this is truly mimicking remote operation. So here you can see there were a couple tries and repositioning of the UAV until they ultimately grasped and dropped on top that ring. So just to give a brief highlight of the results, interestingly, none of the interface views were significantly better than the other, although the pushing task was missed the most frequently, so the most difficult task as expected. And the egocentric view, so having only that eye in hand camera view, resulted in the highest number of missed tasks, and that was also quite expected. When we look at the task completion time and compare that for each condition, we can see again that the pushing task, this third task, had the highest task completion time. It was also the most frequently missed. And again, some really, some difficulty when they only have this view here. You can see that they actually successfully complete the task multiple times, indicating that this view is very useful for this task. But when it comes time to gain that situational awareness and come back and read what's written, they don't even notice that they knocked down the plate multiple times with the manipulator. So it's kind of interesting, and we got really interesting data on what views are valuable for what tasks. Oh, come on. So we also asked participants what did they prefer, and participants usually preferred the mixed or having both views. A subset preferred only to have that exocentric view. When we look at performance, for participants who preferred the mixed or both views in the interface, their preference didn't match up with their performance. Although they liked it better, they actually performed the same with the mixed interface and the exocentric interface. But when we look at participants who preferred only to have that one view, when they used their preferred interface, they completed tasks 50% faster than when they had both views, indicating there are some individual differences in how the information of both views is used. So to understand that, we also collected video and did eye tracking to see at what times during the tasks did they use the exocentric and the egocentric view. So we did this only for that mixed condition. And what we found was that interface preference and view use didn't quite match up. So if you preferred to have both views versus if you preferred only to have one, you were still switching back and forth between the views just as much and watching both views just as much. So this was an indication of perhaps cognitive tunneling, where someone may not want to use or may not find a view useful, but is still allocating attention back to that. So there's definitely from these data a time and a place where certain types of information are valuable depending on the task. And so we're now taking these results and we can use them to inform adaptive interfacing, where depending on the task description and depending on what is shown in each view, we can now have the interface adapt and highlight specific views, depending also on what the user might prefer and depending on what needs to be completed within that remote environment. So why is this important now for ag and biosystems? Well, I think there's a lot of applications in which physical interaction with the remote environment is necessary, and these tasks may not be well defined. So one area where this is actually starting to become more automated is materials and structure inspection, where the environment is more rigid. It's still very dynamic and perhaps unstructured, but the tasks can be more defined. So this is a definite kind of hot area for remote manipulation right now. But I think there's quite a few unexplored areas in agricultural and biological engineering. And just as an example, I have a graduate student now who's working on physically interacting with, again, loblolly pine trees to aid in the automation of controlled pollination. So at seed orchards, workers, you can see here, these aerial work platforms go up and inject pollen into these pollination bags when the trees are flowering in the spring to produce more seed for establishing loblolly pine orchards. While the automation of placing and removing the bags is quite challenging, we're developing a system now that can interact and grab these bags and hopefully ultimately puncture the bag to apply a specific amount of pollen. So taking a blended autonomy approach with the human and the robot to best complete this task is one motivating example of where aerial manipulation can help us in an agricultural specific task. And that was all I had for the seminar today. It looks like there's a little bit of time left for some questions, but thank you so much for your attention.
Thank you, Sierra. It's very nice. Very interesting applications and things. Let me see if there are any questions in the chat. I see a clap from Faiza. Does anybody have any questions? Maybe let me ask one just more out of interest. So what are some of the outdoor phenotyping applications that you're looking at?
Right now, we are doing a bit with loblolly pine in forestry applications. They have some orchards where they have established up to, I think they start making decisions after 5 years of establishment about. So things like stem straightness and height, and also cone counts, production, flowering time are all of interest. So we're collecting preliminary data for some of those traits as well. And we were hoping to do some strawberry phenotyping, but that was right when COVID happened. So unfortunately, that did not take off in the exact way that we wanted it to. And then we're also working a bit on miscanthus, mostly.
Oh yes, that's a very interesting one. Very interesting.
Yeah,
problem.
pretty challenging field conditions for sure.
Yeah, reconstruction is really hard with that. And what are so you mentioned a number of these things. So a number of these things seem like they could be done with cameras, right? And machine vision, or would you need like hyperspectral and?
Yeah, so most of the plant architecture traits, like for biomass and things like stem straightness, yeah, computer vision, also exploring a bit with LiDAR. But yeah, really for the hyperspectral was useful for, we also did a similar study as that cold tolerance study with rust disease tolerance. So we have that study as well with the hyperspectral imaging, but those are more at the greenhouse scale as opposed to the field scale.
Yeah, so I mean, another just question from the vision side, something I'm interested in finding out more in the specialty crops, I guess, that are more open to the sun. Does has the sunlight been an issue in terms of quantifying some of these metrics from vision?
Yeah, that's a good question. So the orchards are kind of a more unique environment. So we haven't had as much of a problem because we actually do conical surveys around some of these trees because they are the seed orchards and the stands for plant breeding. They space the trees out because they want them to like grow to their full potential. So usually there's room to do like conical surveys. And there is some background noise, but things like combining depth imagery with the RGB imagery is useful in those cases. But yes.
Other questions?
I have a quick question. Can you hear me?
Yeah.
I'm wondering if you make one of the saplings tolerant to a minimum of temperature, does that necessarily imply that it can tolerate very rapid temperature swings?
That's a very good question, and it brings up the nuance between cold hardiness and freeze tolerance. So quite often those are used probably incorrectly interchangeably, where you're exactly right. A single freeze may not be the same as a long exposure to some low temperature. And so they are interested in looking at both. So we did this study assuming that freeze tolerance is correlated to, but perhaps not the same as cold hardiness. Cold hardiness studies are a little bit harder to have control over because you need long periods of cold weather, but you can't really stick the seedlings in a freezer for weeks because there's no light. It just gets kind of tricky to do that. So yeah, that brings up a really good point, and it may not be exactly the same, but typically those traits are very much related. I hope that answered your question.
It did.
You mentioned you're using the depth cameras for some work, and how are they performing outdoors? And can you specify the exact one that you're using? Like, we're just starting to use some depth cameras here, and we'd just like to know what's your thoughts on?
Sure. So I've mostly used two, and we have limited data, but one is the Duo MLX. And that, I mean, it has auto exposure. It's been working quite well in outdoor environments. We're using that as our primary depth sensor for the pollination, automated pollination with the UAVs. And then I guess a semi-indoor environment in a different project that I did not discuss, but it's a precision livestock project where we're using the Intel RealSense, and some of the pens are exposed to sunlight, and we actually do find that if there are bright highlights, shadows, that can cause some holes and patchiness in the reconstruction. I mean, that can be remediated in post-processing, but I find that sensors perform a little bit less well in brightly lit conditions. Although I think there are a couple new sensors, also one by Intel, that we have not tried that came out this year. I can't speak to that performance, but I mean, the ones we use are, I think, commonly used by other groups as well.
Yeah.
So,
okay, thanks.
Hope that helps, yeah.
Okay, very cool. So thank you so much, Sierra, for the talk. And I think if people would like to contact you, what email or something they should use?
Oh yeah, I forgot to put that. It's just Sierra Young at NCSU, like North Carolina State University.edu.
Yeah.
So
I'm sure they can find that too on Google. So feel free to ask further questions as necessary. So I really appreciate your time and giving us this very wonderful talk.
Great. Thank you so much for having me.
Yeah, thanks. Yeah. Bye.